Hello to all, greetings to you. Myself, Mr. Karthikeyan, mentor in Botany and Zoology subjects. So, in this video session, I want to explain to you about intermediate public examination question paper from Botany subject year 2021, 2 marks answers. So, here we have 10 2 marks answers which were given in the year 2021 intermediate second year botany subject. One by one answers I shall tell you. Listen carefully and try to learn and grasp the information. First question is, what is the difference between guttation and transpiration? So guttation is different, transpiration is different. So let's see. So first point, guttation, it will occur at night times. Transpiration, it occurs during daytime. Second point, hydatodes involved in guttation process. Stomata are involved in transpiration process. The third point, in guttation, water droplets are released. Water droplets, small water droplets are released. In transpiration, water vapor is released through the stomata. So this is the answer for the question. Write the difference between guttation and also transpiration. The next one, photolysis of water and where it occurs or takes place. In previous videos I have already explained. Many answers were repeated in different years. 5 or 6 or 4 answers, questions, they will be repeated. No problem. All answers I will explain in all videos. Don't worry. So this photolysis already I have explained. Once again I am explaining to you. In chloroplast, on the grana, photolysis of water takes place. So here water molecules will be splitted by the help of photons. That is known as photolysis of water. During this process, oxygen is evolved into the atmosphere. This was first time discovered by the scientist Robert Hill, because photolysis of water is also known as Hill's reaction. Hill apostrophe S, scientist name, Hill's reaction. That's the answer for that question. Third one, conjugation. So from bacteria lesson this question is raised. Conjugation nothing but two bacterium cells, they will exchange their genetic material through direct contact or through the sex pilus or through conjugation tube. That is known as conjugation. First time this was discovered by two scientists. They are Tatum and Liderberg. So Tatum and Liderberg, these scientists, they were discovered this process in E. coli bacterium. Escherichia coli. Short form is E. coli. That is the answer for this question. The next question is, give the example for RNA containing as genetic material viruses. So we have 3 types of viruses. In these 3 types, RNA is the genetic material. What are those viruses? Number 1, poliovirus. Number 2, influenza virus. So one is polio, second one is influenza. So third one is optional one. We may write or we may not write. So that is TMV, tobacco mosaic virus. So, polio virus, influenza virus, tobacco mosaic virus. So, RNA is the genetic material. Then one more, PCR technique. What is mean by PCR technique and where it is used? So, the answer is, the full form of PCR is polymerase chain reaction. So this is a full form, first point. Then second point, this technique is widely used in DNA fingerprinting and DNA technology and to produce desired DNA copies, multiple copies, billions of copies. To produce those copies, PCR technique is widely used. Polymerase chain reaction. That is the answer for this question. Then coming to sixth one, what is GEAC and what is its function? So full form of GEAC is Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. That is the first point. 1 mark for that. The next point, what is the main motto and function or characteristic feature application of this GEAC in the sense? It will guide and control and suggest the DNA technological experiments. So DNA technology related all types of experiments in botany and zoology subjects, these two fields, those all are under the guidelines and control and approval of this GEAC worldwide, because that is Genetic Engineering Approval Committee. That is the answer for that question. Then seventh one, what are fermenters? Where they are used? You knew already fermentation process. So, in industries, large scale fermentation process will be done. In that process, large containers and vessels are used. So those large containers and vessels used in fermentation process known as fermenters. That's it. That's the answer for that question. Then eighth question is, what type of bacterium present in the rumen of kettle? That is a question. The answer is simple. Methanogens bacteria. Methanogens, nothing but methane gas producing bacteria. So the answer is methanogens bacteria. That's it. Then one more, who proposed chromosomal theory of inheritance? The answer is, there are two scientists. Their name is Sutton and Bovary. The scientists Sutton and Bovary, they were proposed chromosomal theory of inheritance. Then last one, xenophore. What is a xenophore from bacteria lesson? Naked the naked genetic material is known as xenophore. The naked genetic material is known as xenophore, which is present in a bacterium. The naked circular DNA structure is known as plasmid. So plasmid in the sense naked circular genetic material DNA, that is plasmid. Naked main genetic material found in or present in bacterium is known as xenophore. So this is the information and the answers of these 10 2 marks answers belongs to 2021 intermediate second year Botany subject from our Andhra Pradesh. Thank you, by the way, myself, Mr. Karthikeyan, mentor in Botany and Zoology subjects, NEET. Signing off.